Hi guys, and welcome to Biological Sciences for Year 10. This is activity 12 where we're going to increase our understanding of DNA, and we're going to try and answer the question, what is the genetic code? Now, what we learned earlier is the idea that we've got DNA. DNA is made up of single nucleotides which are all joined together. It's double-stranded, which means that each nucleotide joins together with bases. The outside rungs of the bases basically are the phosphate groups, and we've got these combination pairs of these bases where we've got adenine going to thymine and cytosine going to guanine. But the question is, how does the DNA actually go and produce a characteristic? Let's face it, this unit has been all about trying to study genetics and how characteristics are passed from one form to the next. So how is it that we've got these sort of bases which can now code for a characteristic? Now, the key thing to understand is that characteristics are set up by the production of proteins. Now, proteins, if you remember back to your junior science, are made up of amino acids. Now, what tends to happen is when the DNA is read, it's read one single strand. So one strand of the two double strands of DNA is going to be read. And when it's read in groups of threes. Now, those threes are called a codon. Now, what happens is, floating around in the cell will be anticodons. Now, these are bits of genetic material which are made up of bases. Now, we have three bases which basically are going to be carrying with them or be attached to, will code for a specific amino acid. Now, we've got something in the region of about 22 amino acids in nature. Now, your amino acids come in through your food. So you take in food, you break it down, the protein is broken down into its constituent amino acids. The amino acids then go to the cell. Each amino acid is then coded for by a specific combination of bases. So, for example, ATC will code for a specific amino acid. ACG may code for another amino acid. And what happens is, as we read along the code of the DNA, reading it in groups of threes, their constituent amino acids are going to, the constituent anticodons, basically what partners join with those three, will come in, pulling with it an amino acid. The amino acids then join together, and when the amino acids then join together, they form a long strand of amino acids. That's your initial, or what we call a primary protein. Once that is being produced, the protein then detaches, and then it starts curling and joining together to form a secondary form of the protein, and then little bits are then added to the protein to make what we call a tertiary protein, which is basically going to code for maybe a specific reaction to occur. It may work like an enzyme, and the result is you get your specific characteristic which forms. Okay, so let's have a look and see how that works. So here we've got a DNA chain which has been produced. So it's one form of the DNA. So to that, what happens is we bring in its constituent anticodon. So the anticodon, the first codon which we read is the first three, which goes TAG. Basically what we've then got is an anticodon which is made up of ATC. Now that is pulled in and produces its amino acid. Now that amino acid in this case just happens to be amino acid number 1. So then we read the next codon. The codon is CTA. So we pull in its anticodon, which is going to be GAT. That comes flying in, and it will bring with it its constituent amino acid. Now in this case it's amino acid number 2. Then it goes along and reads another codon. Now it's a protein that is actually going to be reading this DNA. So it's GCT, its anticodon will be CGA, and it brings in amino acid number 3. The next codon, AGC, brings in the anticodon TCG, amino acid number 4, and then the next codon, TAG, brings in its constituent anticodon ATC. Oh, we come back and we've got amino acid number 1. Now these amino acids now are all joined together, so they bond, they're all close, and there's your protein chain. So what then happens next is once we've got that protein chain, the protein chain then detaches. So that leaves just the anticodons floating around. And you say, well, what happens with those anticodons? Well, what then happens is the first anticodon then comes off, and it will be looking for its constituent amino acid. So it's amino acid which comes from your digestion, comes in and it joins onto it. Then the second one comes off, and its next one, its constituent amino acid joins, and so forth. Each anticodon then comes off, which now leaves us with a DNA chain which is ready to make a brand new protein. So this continues all the time. Now what tends to happen is, and you'll look at this in senior biology in more detail, we don't have the whole of the DNA detaches like a zip and then reattaches. Just a little portion unlocks and it goes along and reads it, and then once it's been read, it then retwists. It joins with its partner strand and then retwists. So you only get a little bit of the DNA which is prized apart. The codons are read, the anticodons come in with their amino acid, then they detach, etc., etc. So you get this protein which is built up on a continuous basis, and this reading protein just goes along the whole DNA strand, adding amino acids and then detaching. So we don't need to know that in any great detail. You just need to be aware that we build up these anticodons which join in, which are literally their corresponding pairs. Okay, well, I hope you found that useful. It's a little bit complicated to try and understand. I suggest you watch it, the video maybe again, to try and understand exactly what's going on. But at this level, all you need to understand is the idea that you get your chain, you get a corresponding strand, they're read in threes, and they bring in an amino acid, which joins together to make a protein. That protein detaches, leaves the cell, and that protein is responsible for our characteristics. Okay, well, I hope you found that useful. Join me again, and we'll talk about what happens if we have a change in our genetic code. Okay, thanks for watching.